are not from one of these three groups, please find another hall. If you are, I hope you like your seat. Okay, well, not too many people getting up. That's good. That means we can get started. We're a little late, so we'll get right to it. My name is Shai. I have a graduate student here at the Hebrew U. These days I'm graduating from my PhD studies in biology in the genetics department. I grew up at the Hebrew U. I did my bachelor's and master's degrees in Mount Scopus, the campus you were at yesterday, last night. I studied law, and after a short legal career, I came here and pursued my passion to become a scientist. I've been doing that for the past decade, here in the Givat Rav campus. Okay. Today, I am the director of the Joseph Meyerhoff Youth Center for Advanced Studies, which means I see a lot of youth when I come to work. I deal a lot with education, science education. I like it very much. And today it is my pleasure to present to you one of the most distinguished researchers and scientists at the Hebrew University, Professor Howard Cedar. The CV of which you have in your book, so I will not repeat it. See where he studied. He studied in the United States, and came and started a very successful scientific career here in Israel at the Hebrew University. And he's going to talk to you today about DNA methylation, which is a part of a vast field called epigenetics, which I had the pleasure of studying as well. So, without further ado, Professor Cedar, thank you.
Good afternoon. Welcome to everybody who came from very far away, which is a lot of you. And welcome to all those who came from very close. It's very nice to be here today. And I'd like to share with you some of the scientific adventure that went into what you're going to see is what the beginnings of epigenetics. Okay. I'm going to start off very simply, and hopefully we'll get into the subject and be able to see the sophistication of what goes on in the control of our genes, the control of the genes of the human being, and how this control, this regulation, is responsible for the development of man. Okay, so you all know that the body is made up of organs. The organs are all made up of cells. Each cell, by the way, is basically a mini organ. So if you look inside the liver and you take an individual cell, that individual cell is capable of doing everything that a liver does. Okay? So every organ is made up of these subunits called cells. And within the cells, what defines the cells, what defines their shape, their color, their position, and their function are proteins. Proteins are basically the Lego of life. Just like if you want to build a bridge, you need building materials. If you want to build an organism, you need proteins. Those are the building materials of life. Okay? Proteins, I'm sure you all know and recognize them. I'll just give you a couple of examples. Unfortunately, I don't have a pointer, so it might be, I might have to do a little bit of a dance here. But I'm sure it'll be okay. So what you see here is the iris of an eye, which in this case is blue. Some of us have brown eyes. And of course, the iris is made up of cells. Excuse me. Iris is made up of cells. And within those cells are many proteins. One of the proteins is this blue color. This is a pigment. It's really a protein. Okay, so that's one example of a protein that gives a quality of color. Okay, let me give you another example. The thing on the left is a red blood cell. Oh, wow. Okay. This is a red, well, modern technology. This is a, maybe, hello? Maybe you can also get something that switches the slides. Never mind, it's okay, we'll manage. Thanks a lot. Okay, so this is a red blood cell. It's red. as you all see. And it's major protein in this cell that makes up almost 95% of all the building blocks of this cell is one protein called hemoglobin. It's a complicated protein. It has subunits, a number of different molecules. Nonetheless, it's a protein. These blue things are molecules of iron. That's actually what gives it this color red. And this is another kind of protein. This kind of protein is not structural. It doesn't give color. It does give a little bit of color. But what kind of protein is this? This is a protein that gives function. You all know that hemoglobin is responsible for carrying oxygen around the body. These red blood cells go to the lungs. In the lungs, they meet oxygen. The hemoglobin is responsible for grabbing the oxygen. It then holds on tight to the oxygen. The red blood cell gets to the periphery of the body, all the little vessels of the body. And once it gets there, it opens, releases the oxygen so that the oxygen can be used all over the body. So this is a protein that has function. It can move, it can change shape, it can recognize protein. Many enzymes in the cell are responsible for running the metabolism of what goes on inside the cell. These are all proteins that provide function. These are proteins that grab one molecule like sugar, are able to take it, break it up, make other molecules so that those molecules can be used to construct other things within the body. This is a metabolism. Okay? There's another kind of protein. Another kind of protein is a structural protein. This is actually a group of proteins put together, but it's easy to see from the shape of this protein what it does. You notice this protein actually looks like a tunnel. It has a hole in the middle, and this protein is actually a tunnel. It's a channel. This is a protein that's located in the membrane of cells. Okay, so here's the membrane, and in the membrane there's this channel. And the channel is responsible for passing things in and out of the cell. Okay? Note that the shape of the protein determines the shape that determines its function. In the same way, for instance, antibodies. Antibodies have to recognize a whole slew of different kinds of molecules in our environment, within our body and within the environment. And that they do, those are all proteins, and all proteins with a different shape. And each one of those proteins is able to recognize the molecules we use for smelling. There are over 1,000 different receptors for smell in the nose. Each one has a little different shape that recognizes a different smell in our environment. The smell of fruit, the smell of alcohol, the smell of things that don't smell so nice as well. All this is carried out by proteins. Okay? Now, where do these proteins come from? Well, first of all, where do these proteins come from? It turns out that these proteins, there are about 50,000 of them in the human. These proteins have to be made in all of these individual cells. And in order for them to be made, there has to be a factory for making them. And there is a factory. A number of years ago, Ada Yonat received the Nobel Prize. Ada Yonat is a scientist from the Weizmann Institute here in Israel, in Rehovot. She received the Nobel Prize for figuring out the structure of this factory called the ribosome, which makes all these proteins. But factory is not sufficient. You need instructions. Each one of these proteins is different. So there must be some place in the cell where there are instructions for how to make all these different 50,000 proteins. And those instructions are basically what we call the genetic material. In every cell there's like a book that has the information, has the instructions, how to make the proteins that make up the building blocks of the cells that make up the human body. And it looks something like this. This book, this genetic information, has a language. Like any book, there has to be a language. This language only has 4 letters, and we call them A, T, G, and C, the 4 letters. Obviously, these are chemicals. Okay? Each chemical with a little different shape, but we as scientists give them names. Obviously, the people who discovered it, the native language was English, so they gave them the names in English, A, T, G, and C. And a typical region of this book looks something like this. It's a string of letters. But with these letters make up words. Each word is 3 letters. And a whole bunch of words together make up a sentence. And each one of those sentences basically is the information for making a protein. And it works something like this. So this is we're looking at a book now. And now the cell is going to read this book. So it starts off, it reads ATG. ATG is a code for taking one amino acid. You know the amino acids, you know many of them: lysine, threonine, serine, phenylalanine. These are all names that you've heard. These are all amino acids. Okay? So this is the code for an amino acid. Proteins are a string of amino acids. So first of all, it reads ATG, puts this amino acid, which we'll call red. It could be tyrosine, phenylalanine, or maybe somebody knows the code to ATG. What is it? Methionine. Methionine, maybe? I don't know. I didn't do it accurately, so I'm not sure. Okay. Then it goes on and reads the next word, 3 letters, CTA, adds the next amino acid, which is called blue. Then GAT, next amino acid, CAT, next amino acid, CGG, next amino acid, TCA, next amino acid, CTA, next amino acid, until we've made a protein. Okay? Every protein is different because every protein is made up of different amino acids in a different order. And it's this order of those amino acids that gives the protein its quality. Okay. That's a little bit too dark, I think. Okay. I can't even see the how to make it go forward. Oops. I can see I made it go backwards. That can also happen, by the way. Proteins can get degraded, even on the computer. Okay. Now what happens to these? Here's a recently synthesized protein. In other words, this was just read. This booklet, by the way, is located in the nucleus of the cell. The cell read this information, made this string of amino acids, and now because of the properties of each of the different amino acids, this string now folds on itself. Okay? You know, some of these amino acids have positive charges, some have negative charges, some have certain shapes, some have different shapes, and because of that, it folds in a certain manner, and it makes a protein. And this protein now has a structure, thank you, now has a structure, okay, which will give it a certain function. It can be a structural protein, making the cell a certain shape. It can be a protein that gives color. It can be a protein that gives function. But it's all determined by the order of these amino acids, which is, from the beginning, determined by what's written in the DNA. By the way, as much as we know about biology, the ability to read the DNA, we can do. And we, as people, can read this, or every one of you could read this DNA and decide what will be the string of amino acids. But we still don't know the code of how to translate that simple linear information into what will be the ultimate shape. It's still one of the most important codes of biology that we still don't know how to do. In any event, it's read. The string of amino acids is made, it makes a protein. The protein now has structure. There are many of these sentences in the genome. Okay? They're located on the DNA. So here's a sentence on the DNA, like similar to the one we just read. And of course, not only is there letters and words and sentences, but actually there are also volumes of books. And we call those volumes, here's one volume, we call it a chromosome. Okay? And actually in every cell there are many chromosomes, and every chromosome has a bunch of different genes. Each gene codes for, is the information for, an individual protein. Okay, we've gotten, this is just to give you a feeling for the size of this book, a size of this genome that we call the full DNA, our full DNA, which by the way today we're capable of taking human DNA and determining the sequence of all of the letters from the beginning until the end. How many letters? There are 3 billion letters in every single cell. Every one of these books has 3 billion letters. Just to give you a feeling for what this means in terms of information, a single page of a book has about 3,000 letters on it. 3 million letters would be a volume of a book with 1,000 pages. 3 billion letters is 1,000 of these books, which if stacked up would reach about 50 meters high, and that's what we have in every cell of the body. So it's a large, large piece of information. Okay, now the DNA is, this booklet, is located in every cell of the body. How does this come about? It comes about because when each of us is conceived, we receive half of our genetic information from our mother, from the oocyte, from the egg. We receive half of our genetic information from the sperm, from the father. That generates a cell. That cell has all the information in it. That's the first cell of the body. It's called the zygote. In order to make an organism, that cell has to start dividing and making many, many, many cells. For instance, in our bodies, we have something like 10 to the 15th cells, which in terms is about 1,000,000 billion cells in our body. And they all came originally from the first cell. The first cell divides, makes 2 cells. Those 2 cells divide to make 4, to make 8, 16, etc., until it makes up the entire body. Now before the cell divides, it does something very important. It copies the booklet. So it now has 2 copies of the book. One it gives to one cell, one to the other cell. Then each one of those divides, but before it divides, it has to copy all the information. This, by the way, is not a simple task. You can ask yourself, if I would give any one of you to sit down and copy a page of a book, which is 3,000 letters, I guarantee you that every single one of you would make at least one mistake. And every one of our cells has to be capable of copying 3 billion letters in a period of about 8 hours and not make many mistakes. It makes mistakes, but it makes very, very, very few mistakes. And a very important part of biology, fidelity. Okay, every cell of the body has the same book, which we got from our parents. But now I'm going to show you something about cells, which you all know, but I want to put emphasis on it. So this is an example of a cell. This is not its real color. It's not necessarily green. This is artificially stained. And I'm sure all of you can probably tell me what kind of cell this is. What kind of cell? This is a neural cell. This is from the nervous system, from the brain. Okay? It function. You can see its shape fits its function. This cell has to be capable of talking to other cells, and it does that through these many, many branches. Okay? Some of them are axons, some of them are dendrites. They stretch out and they can talk to other cells. This cell, you don't see all of the enzymes in this cell, all of the proteins in this cell that are able to make the transmitters, which are used for actually talking between the different cells. There are also proteins in this cell that are specific to the nervous system that allow it to make this shape. Okay? There are also proteins that are involved in getting this cell to be in the right place in the body. All of those are specific proteins that are needed for this cell, for this nerve cell. Now this is a nerve cell, but not all the cells look like that. Here's an example of cells that don't look like that. Who wants to guess? It's very easy, it's obvious.
White cells?
No, not a white cell.
Fat cell.
Fat cell, very good. This is a fat cell. And the cell, here's the periphery of the cell. These things are the nucleus of the cell. And this are just plain globs of fat. Okay? Now obviously this cell must use much different proteins than the nerve cell. It has a different shape, it has a different color, it has to be able to metabolize the fat, to make the fat, to digest the fat. It uses much different proteins. So here's a cell, also a cell, has the same information, but it's using much different proteins. By the way, when this person here, who I would guess is quite fat, from the amount of fat that he has in his cells, when he loses weight, he'll still have the same number of cells, but they'll all be a lot smaller. They'll be more like this. Okay, here's another type of cell. It's a little harder to guess. This is actually a liver cell. These are liver cells. Again, you notice, first of all, liver cells have about 1,000 different specific enzymes that are involved in detoxifying blood. They're only found in the liver. They're not found in any other place. But the information for making those proteins is obviously in every cell of the body. Okay? What kind of proteins? You see this structure, the way this cell is touching, kissing its neighbors? That involves proteins. In order for cells to be able to be in close contact with each other, it involves specific proteins. They have a specific shape. That also involves specific proteins. Okay? So here too, there are specific proteins that are made. And the question that I want to ask, and this is basically going to be the core of what I'm going to tell you now, is how does it work? How can it be that every cell of the body has the full encyclopedia of information, how to make every protein that could possibly be, all 50,000 proteins, yet each particular cell seems to be picking and choosing specific regions of that DNA to read in order to make specific proteins needed for that particular cell. And one of the keys, probably the main key in animal cells, is that the text that we've been talking about up till now, the text of the book, can actually be modified. And it's modified by a simple chemical modification called DNA methylation. Only the C, the letter, the chemical letter that we call C, only it can be modified by methylation. Okay? Methyl, for those of you who are not chemists, methylation is a chemical group. It covalently attaches to the C. It's a real modification. It doesn't affect the text. The text remains the same. I still know this is C. But it has like a flag on it now. It's been modified, a cap. And every place where this cap sits serves as a signal to say, don't read me. I'm not readable. Places that are unmethylated, that don't have the methyl, are readable. Places that have the methyl are not readable. So just to give you a feeling for how it works, this, by the way, is what C looks like chemically, and it can be methylated. In human beings, this is the only letter that's methylated. It's methylated with a methyl group, carbon H3, hydrogenation. So now, if we with this new knowledge that not only is there a text, but there's also an annotation of the text called methylation, we can now look anew in individual tissues. So what we're doing here is looking at the gene for blue eye color, the gene for eye color, the pigment gene. This is it. Okay, this is the text of that gene in the book. This text is exactly the same in the iris of the eye, in liver cells, and in nerve cells. But now if we look at the annotation of this gene, we see that the annotation gives information on how this gene, how this information is read. In the iris of the eye, it's not methylated. It can be read, and as a result, it makes the pigment that we see here in the iris. On the other hand, the liver has the same gene. Maybe surprisingly or not surprisingly, the liver is not blue. And the reason is because this gene, this piece of information, is not read in the liver because it's methylated. And in nerve cells, it's also not read. So it's only read in the eye, and that's the reason why eye color only appears in the eye, in the iris of the eye. Another example, this gene is for the protein albumin. Albumin is the most abundant protein in the body. It's made in the liver. The information for making albumin is in every cell of the body. But if we now look at the annotation, the footnotes, we see that albumin is unmethylated in the liver, and it is indeed expressed here, but it's methylated in the eye, methylated in the nerve cells, and methylated essentially in every other tissue, every other cell of the body. Okay? So DNA methylation serves as an annotation and a mechanism for regulating the expression, the reading of genes. Inside the cell, if we were to look at the reading machinery, this reading machinery in the cell, by the way, the deciphering of the structure of the reading machinery was done by another Nobel laureate, Roger Kornberg, who you're going to hear in another 2 days. Right? He was the one who figured out what this reading factory looks like. Okay? You'll get to that. Anyhow, how does it work? The reading machinery goes along the book to read. Here it can read this gene, this piece of information, because it's not methylated. But when it gets to this gene, because of the methylation, it's not able to read. And as a result, this gene is not made into a protein. It goes on, it reads over here. Again, this can be converted into a protein. Okay. Now, I just want to give you a feeling here for how this works. What we've done up till now is looked at tissues. So you look at the liver, you see this is methylated, this is not methylated, and it's clear to us now that there's a methylation pattern in the liver. So some regions are methylated, some regions are not methylated, and that will help determine what will be read and what will not be read. If we look into a different tissue, we'll see the same text, but now the annotations will be in different places. And again, like switches, some will be on, some will be off. But if we look at this from a developmental point of view, the picture is a little bit different, a little bit more poignant. So we start off in early development, as you'll soon see. We start off in early development, all of the methylation is erased. Okay? So any methylation that came from the sperm and the egg, all erased. And now, by the way, those cells are stem cells. Now, there's a program for methylating, which we're going to go into this a little bit in a minute. But what I want to point out is that it's the placing of these methyl groups that actually determines differentiation, that determines the building of different tissues. This region of the embryo became a liver because the genes were regulated. Methyls were put here, methyls were taken off here, and to generate exactly the pattern that you need in order so the right things will be read to make a liver. In another region of the embryo, okay, the periphery of the embryo, these switches are set in such a way such that the periphery will become the skin and the nervous system. Okay? So it's not that the liver has regions that are methylated and unmethylated, or that the skin has regions that are methylated or not methylated. collection of prayers. But notice this prayer book also has something else. It has this. This is also written in the same letters. But it's not a prayer. This is instruction how to read the prayer. And what this instruction says is that the following prayer is read every day of the year, except for 2 different days. On those 2 days, you don't read this. So within this book, not only is there structural information, information for making proteins, information for praying, but there's also information using the same letters for how to read it. And that's exactly what happens with methylation. So, here's a piece of the DNA. Okay, imagine that this is the whole genome. These are genes, sentences that make proteins, yellow. Next to many, many genes, perhaps all the genes, also in letters, there's another sign language here. It's like a flag saying, what should you do with this gene? How the gene should be read. So for instance, this gene, and this gene have a red flag in front of it. This red meaning it's some sequence of letters. There's a red flag in front of it. What does that mean? That means that those genes, at the time of implantation, when everything gets methylated, those are the ones who are going to get methylated. And the ones that have the red flag are the ones that are protected. Why are they written into the book already? It's already written into the book. How to read is already written into the book. Okay? Now, there were more signs here. So this gene has a blue tag. This gene has a pink tag, purple tag. This gene has both a blue tag and a purple tag. This is information for when it should undergo demethylation. Okay? So now, if this will undergo demethylation in a blue cell, a liver, or a kidney, or a skin, this will undergo demethylation in a purple cell. So now, if you look at the two different cells, you see a blue cell, okay? All of the housekeeping genes unmethylated because of the red signal from the beginning. And notice that this one is unmethylated only in a blue cell, because it had a blue tag. This didn't have a blue tag. This had a blue tag. So in the purple cell, it doesn't get unmethylated. This one had a purple tag. So it gets unmethylated in the purple cell, but doesn't get unmethylated in the blue cell. And this gets unmethylated in both. Okay? So the information for where to put the methylation, where to take off the methylation, is all written in the original book. With that, I'll stop. If you have any questions, I'll be glad to answer.
Thank you very much. Tomorrow there will be a follow-up discussion about this stuff, but we do have a few minutes, so we will ask the questions.
Okay, if you come tomorrow, I promise to tell you in brief. Okay? That wasn't meant to be an application. Not a lot of it. Okay. So, cancer cell started off as a normal cell, and it became cancerous. I won't go into detail about how that happened. A lot of that has to do with changes in the text. Okay, the DNA itself, the text. Okay? But if you take a look at a cancer cell and compare it to a normal cell, a cancer cell does a whole bunch of things all wrong. Okay? It doesn't sit nicely where it's supposed to. It divides too rapidly. It doesn't metabolize it. Many times it breaks away and attacks on the tissue. Many times it goes in the blood. It has the ability to take off home in another place in the body. Normal cell will never do that. It has different properties. What does that mean to you? It has different properties. It's making different proteins, different building blocks. Why? that, be able to change the methylation. We'll talk about that a little bit. Yeah, more.
During cell differentiation time, how does the cell know, as in which of the genes should demethylate?
Yeah.
How does it know which of the particular ones should go for?
That's a good question. This is a developmental question, which is interesting in itself. Okay? How, when you start off with this nice, round, perfectly symmetrical embryo, right? Why is the liver come over here, and the skin over here, and the kidney over here? Well, how does that happen? So it turns out that even though the embryo is symmetrical, it sits in one place in the uterus, and that makes it asymmetrical. So now every part of the embryo is exposed to different things. Some of them are close to the point of implantation, some of them are up, some of them are down, some of them on the side, and as a result they're exposed to different things in the space of the embryo. And that sets in motion changes in the cell which help determine what's going to be liver, what's going to be skin.
And I think the mother's egg cells, the mRNAs that are formed in mother's egg cells also affect that thing.
I don't know. It's a good question.
mRNAs that are formed in mother's egg cells.
No, I understood the question. I'm saying it's a great question.
No, I'm just answering. Not answering you.
Yeah, in some organisms, right. In some organisms, already, if you take a frog, for instance, a frog egg, it's an amazing thing. Anybody here ever see a frog egg? In nature? Yeah. If you look, if you go to a pond, the right time of year, you see these beautiful eggs, big, very big, and you pick one up, and the first thing that strikes you is that they're asymmetrical. The top, one half, has a dark color, right? Another half has a bright color. One cell is asymmetrical. And if you now look inside that cell, at the different parts of the cell, there's some RNAs only the top part and some RNAs only the bottom part. Already at the beginning is asymmetry. And that asymmetry can help this egg develop its different organs because each part of the cell is basically different.
Last question. There.
Is there any disease that come from misplaced
Start at the beginning again.
is there any disease that come from misplaced methylation in the DNA? Disease.
Oh, yeah, good question also. There are a number of different diseases where the major cause, the major bottom line cause of disease is DNA methylation. One example is a disease called Fragile X syndrome. Fragile X syndrome is the most common cause of mental retardation all over the world. Okay? And it's basically caused by the inactivation of a gene by DNA methylation. Thank you.